Welcome to this tutorial on biochemistry of cancer, brought to you by Medical Biochemistry Power Capsule series. This video covers the following CBME curriculum topics: describe cancer initiation, promotion, oncogenes, and oncogene activation. Also focus on p53 and apoptosis. Describe various biochemical tumor markers and the biochemical basis of cancer therapy. Cancer is a complex group of diseases characterized by the abnormal growth of cells that have the ability to invade and destroy normal body tissues. It can arise from various factors, including genetic factors, environmental factors, and lifestyle choices, and can affect virtually any part of the body. Cancer poses a significant health problem and burden due to its pervasive nature and devastating impact on individuals, families, and societies. Some of the most prevalent cancers worldwide include esophageal cancer, head and neck cancer, lung cancer, breast cancer, colorectal cancer, prostate cancer, stomach cancer, liver cancer, cervical cancer, thyroid cancer, bladder cancer, and non-Hodgkin lymphoma. There are of course wide variations depending on geography and other factors. Cancer is a formidable foe due to its ability to infiltrate and disrupt the body's natural processes. Its danger lies in its unchecked growth, spreading like a rogue force within the body. Often undetected until advanced stages, it hijacks normal cellular functions, evading the immune system's surveillance and resisting conventional treatments. Cancer is a disease of the genome, irrespective of the cause. The common event always is gene mutation. Malignant cells possess several key properties and characteristics that distinguish them from normal cells. These properties include uncontrolled growth. Malignant cells divide rapidly and uncontrollably, leading to formation of tumors, invasion and metastasis. They have the ability to invade nearby tissues and spread to distant organs through the bloodstream or lymphatic system, forming secondary tumors. Resistance to cell death. Malignant cells can evade programmed cell death mechanisms, such as apoptosis, allowing them to survive and proliferate despite adverse conditions. Genetic instability. They often harbor genetic mutations or abnormalities, resulting in genetic instability and heterogeneity within the tumor cell population. Immune evasion. They can evade detection and destruction by the immune system, enabling them to escape immune surveillance and continue to grow unchecked. Altered metabolism. Malignant cells often exhibit changes in their metabolic pathways, favoring processes that support their rapid proliferation and survival. Tumor microenvironment interaction. They interact with the surrounding microenvironment, influencing immune cells, fibroblasts, and other components to create a supportive niche for tumor growth. Angiogenesis. Malignant cells induce the formation of new blood vessels, ensuring a steady blood supply to the growing tumor, which is crucial for its sustenance and expansion. These properties collectively contribute to their aggressive behavior and ability of malignant cells to evade treatment. Key molecular mechanisms that are involved in cancer include the activation of oncogenes, inactivation of tumor suppressor genes, evasion of apoptosis, and genomic instability. Hence, in the next section, I shall be sharing pertinent information about oncogenes, tumor suppressor genes, and apoptosis. Oncogenes. What are they? How do they cause cancer? Oncogenes are the mutated or abnormally expressed versions of normal genes known as proto-oncogenes. Proto-oncogenes encode proteins that regulate normal cell growth, differentiation, and survival. When these genes are altered, they become oncogenes and contribute to the development and progression of cancer. There are mechanisms by which proto-oncogenes convert to oncogenes, or in other words, oncogenes are activated. Point mutations, gene amplification, chromosomal translocations, and insertion mutagenesis. It's important to remember that proto-oncogenes do not cause cancer, their mutated versions do. Point mutations. A single nucleotide change in the DNA sequence of a proto-oncogene can result in a constitutively active or hyperactive protein. Let me mention an example. The RAS gene family encodes small GTPECs involved in cell signaling. Point mutations in RAS genes lock the RAS protein in an active GTP-bound state, leading to continuous cell proliferation and survival signals. Mutations in RAS are found in various cancers, including pancreatic, colorectal, and lung cancers. Please go through the flowchart to understand better. Gene amplification. The increase in the number of copies of a proto-oncogene can lead to overproduction of its protein product, thereby promoting unregulated cell growth. As an example, we may cite the HER2/neu gene. HER2/neu is a receptor tyrosine kinase gene. Amplification of HER2 results in overexpression of the receptor, which drives aggressive cell proliferation. HER2 amplification is observed in approximately 20 to 30% of breast cancers and is associated with poor prognosis. Chromosomal translocations. Rearrangement of chromosomes can place proto-oncogenes under the control of strong promoters or enhancers or create fusion genes with novel properties. The example of BCR-ABL may be cited. The BCR-ABL fusion gene is created by the Philadelphia chromosome translocation in chronic myelogenous leukemia. The resulting BCR-ABL protein has constitutive tyrosine kinase activity, leading to uncontrolled cell division and survival. Insertional mutagenesis refers to the insertion of foreign DNA into the genome, potentially disrupting normal gene function. Viral integration near proto-oncogenes can lead to their activation by viral promoters or enhancers. One example is seen in some cases of leukemia, where retroviruses, like human T-cell leukemia virus type 1, can integrate into the host genome, leading to uncontrolled cell proliferation. It activates the proto-oncogene that encodes for platelet-derived growth factor. This is one of the ways by which viruses can cause cancer. Of course, not all viruses are oncogenic. What are the functional consequences of oncogene activation? The consequences include uncontrolled cell proliferation, inhibition of apoptosis, enhanced angiogenesis, and invasion and metastasis. Oncogenes are critical drivers of cancer development and progression. They arise from mutations or alterations in proto-oncogenes and lead to uncontrolled cell proliferation, resistance to apoptosis, enhanced angiogenesis, and increased invasiveness and metastasis. Uncontrolled cell proliferation. Oncogenes often encode proteins that directly promote cell cycle progression and proliferation. The MYC gene encodes a transcription factor that regulates genes involved in cell growth and division. Overexpression of MYC, due to amplification or translocation, drives excessive cell proliferation in various cancers, including Burkitt lymphoma and neuroblastoma. Inhibition of apoptosis. Oncogenes can encode proteins that inhibit programmed cell death, allowing cancer cells to survive and accumulate further mutations. The BCL2 gene encodes an anti-apoptotic protein. Overexpression of BCL2, due to translocation in follicular lymphoma, prevents apoptosis, leading to the survival of cells that should normally undergo programmed cell death. Enhanced angiogenesis. Oncogenes can promote the formation of new blood vessels to supply the growing tumor with nutrients and oxygen. The vascular endothelial growth factor gene is upregulated by oncogenes like RS and MYC. Increased vascular endothelial growth factor expression stimulates angiogenesis, supporting tumor growth and metastasis. Invasion and metastasis. Oncogenes can encode proteins that facilitate tumor cell invasion and metastasis. The MET gene encodes a receptor tyrosine kinase involved in cell motility and invasion. Mutations and amplification of MET are associated with increased invasiveness and metastasis in cancers, such as renal cell carcinoma and non-small cell lung cancer. In the subsequent section, I shall be sharing relevant information about tumor suppressor genes. Tumor suppressor genes are crucial components of cellular regulatory mechanisms that inhibit uncontrolled cell growth and division. They play a key role in maintaining genomic stability, regulating the cell cycle, promoting apoptosis, and repairing DNA damage. When these genes are inactivated or lost, cells can grow uncontrollably, leading to cancer. The important functions performed by tumor suppressor genes include regulation of the cell cycle, DNA repair, induction of apoptosis, and inhibition of cell migration and invasion. Regulation of the cell cycle. Tumor suppressor genes ensure that cells do not divide uncontrollably by regulating key checkpoints in the cell cycle. The ARB1 gene encodes the retinoblastoma protein, which controls the transition from the G1 phase to the S phase of the cell cycle. When retinoblastoma protein is active, it binds to and inhibits E2F transcription factors, thereby preventing cell cycle progression until the cell is ready to divide when the time is right. Mutations obviously lead to uncontrolled division. DNA repair. Tumor suppressor genes are involved in detecting and repairing DNA damage, maintaining genomic integrity. The BRCA1 and BRCA2 genes encode proteins that are critical for the repair of double-strand breaks in DNA through homologous recombination. Mutations in BRCA1 or BRCA2 lead to impaired DNA repair and increased risk of breast, ovarian, and other cancers. Induction of apoptosis. Tumor suppressor genes can trigger programmed cell death in response to cellular stress or DNA damage, eliminating potentially cancerous cells. The TP53 gene encodes the p53 protein, which activates the transcription of pro-apoptotic genes in response to DNA damage or other stress signals, leading to apoptosis. Mutations in TP53 cause failure of apoptosis due to defective p53 protein. Inhibition of cell migration and invasion. Tumor suppressor genes can also prevent cells from acquiring invasive properties and spreading to other parts of the body. The CDH1 gene encodes ecoadherin, a cell adhesion molecule that helps maintain proper cell-cell adhesion and tissue architecture. Loss of ecoadherin function can lead to increased cell motility and metastasis. There are proteins, encoded by tumor suppressor genes, which prevent cancer demons from spreading. What are the mechanisms involved in tumor suppressor gene inactivation? These are mutations, chromosomal deletions, loss of heterozygosity, and epigenetic silencing. Point mutations, insertions, deletions, and other alterations can inactivate tumor suppressor genes. Mutations in the TP53 gene can lead to the production of a dysfunctional p53 protein, which fails to induce cell cycle arrest or apoptosis in response to DNA damage. Chromosomal deletions. Large deletions involving tumor suppressor genes can result in their complete loss. Deletions on chromosome 17p often involve the TP53 gene, leading to its loss and contributing to various cancers. Loss of heterozygosity. Often, one allele of a tumor suppressor gene is mutated or deleted, and the remaining functional allele is lost or inactivated, leading to a complete loss of tumor suppressor function. In hereditary retinoblastoma, individuals inherit one mutated copy of the ARB1 gene and often lose the second copy through loss of heterozygosity mechanism in retinal cells, leading to tumor development. Epigenetic silencing. Tumor suppressor genes can be inactivated by epigenetic modifications, such as DNA methylation or histone modifications, which silence gene expression without altering the DNA sequence. Hypermethylation of the promoter region of the CDKN2A gene, which encodes the p16 protein, can silence its expression, leading to uncontrolled cell cycle progression. Tumor suppressor genes play a vital role in regulating cell growth, maintaining genomic stability, and preventing cancer. Their inactivation through mutations, epigenetic changes, or chromosomal deletions can lead to uncontrolled cell proliferation, genomic instability, evasion of apoptosis, and increased invasiveness, all of which contribute to cancer development and progression. The consequences of mutations in tumor suppressor genes include uncontrolled cell proliferation, genomic instability, evasion of apoptosis, and increased invasion and metastasis. Uncontrolled cell proliferation, loss of tumor suppressor function, removes critical checks on the cell cycle, allowing cells to proliferate uncontrollably. Genomic instability, inactivation of DNA repair genes leads to the accumulation of mutations and chromosomal abnormalities, driving cancer progression. Evasion of apoptosis. Without functional tumor suppressor genes, cells can evade programmed cell death, even in the presence of significant DNA damage or other cellular stresses. Increased invasion and metastasis. Loss of genes that regulate cell adhesion and migration can lead to increased tumor invasiveness and the spread of cancer to distant sites. Examples of T-tumor suppressor genes. TP53, known as the guardian of the genome, P53 plays a central role in preventing cancer by inducing cell cycle arrest, DNA repair, and apoptosis in response to cellular stress and DNA damage. Mutations in TP53 are found in over 50% of all human cancers. Retinoblastoma protein, the RB1 gene product, known as the retinoblastoma protein, regulates the cell cycle and prevents uncontrolled cell proliferation. Inactivation of RB1 is a crucial loss and is associated with retinoblastoma, osteosarcoma, and other cancers. Adenomatous polyposis coli. The APC gene product is involved in regulating cell adhesion and signaling pathways that control cell growth and apoptosis. Mutations in APC are a primary cause of familial adenomatous polyposis and are frequently found in colorectal cancers. BRCA1 and BRCA2. These genes are essential for the repair of DNA double-strand breaks. Germline mutations in BRCA1 or BRCA2 significantly increase the risk of breast, ovarian, and other cancers. What is apoptosis, and why apoptosis is so important for our survival? Apoptosis, or programmed cell death, is a critical process that enables the body to maintain cellular homeostasis by eliminating damaged, unnecessary, abnormal, or potentially harmful cells. It is an essential mechanism for preventing cancer, as it ensures that cells with damaged DNA or other cellular defects do not continue to proliferate. Apoptosis is a crucial mechanism for preventing cancer. So, what is achieved through apoptosis? Apoptosis is a DNA damage response. Apoptosis is required for cell cycle control. Elimination of oncogene-activated cells is through apoptosis, and also tumor suppression. DNA damage response. Apoptosis is triggered in response to significant DNA damage. Cells that fail to repair such damage effectively undergo apoptosis, preventing the propagation of mutations that could lead to cancer. The P53 tumor suppressor protein plays a crucial role in inducing apoptosis in response to DNA damage. When DNA is damaged, P53 can activate the expression of pro-apoptotic genes, such as BRX and PUMA, leading to cell death. Cell cycle control. Apoptosis acts as a counterbalance to cell proliferation. Under normal conditions, cells that proliferate inappropriately or excessively are eliminated via apoptosis, maintaining tissue homeostasis during development. Apoptosis removes excess or improperly developed cells, ensuring proper tissue and organ formation. Elimination of oncogene-activated cells. Oncogenes that drive excessive cell proliferation can also induce apoptotic pathways as a protective mechanism. Cells that overexpress oncogenes often activate apoptosis to prevent tumorigenesis. The overexpression of MYC oncogene can induce both cell proliferation and apoptosis. When apoptotic pathways are intact, MYC-driven cell proliferation is kept in check by simultaneous induction of apoptosis. Tumor suppression. Tumor suppressor genes, like TP53, promote apoptosis in cells with abnormal growth signals or extensive DNA damage. Loss of function in these genes often results in the failure to undergo apoptosis, contributing to cancer development in many cancers. Mutations in TP53 result in a defective P53 protein that cannot induce apoptosis, allowing damaged cells to survive and proliferate. This has already been discussed previously in this video in connection with tumor suppressor gene function. What are the mechanisms by which cancer cells evade apoptosis? Inactivation of pro-apoptotic pathways. Cancer cells often acquire mutations in genes that promote apoptosis. Mutations in TP53 impair its ability to induce apoptosis in response to cellular stress. Downregulation of pro-apoptotic proteins. Cancer cells may downregulate the expression of pro-apoptotic proteins, such as BRX, BRK, and PUMA, reducing their susceptibility to apoptosis. Upregulation of anti-apoptotic pathways. Many cancer cells overexpress anti-apoptotic proteins, such as BCL2 and CO1, which inhibit the intrinsic apoptotic pathway by sequestering pro-apoptotic proteins. Overexpression of BCL2 is commonly observed in follicular lymphoma and helps cancer cells resist apoptosis. Alteration of death receptor signaling. FAS receptor and ligand alterations may occur. Cancer cells can alter the expression of death receptors, such as FAS or their ligands, impairing the extrinsic apoptotic pathway. Loss of FAS receptor expression allows cancer cells to evade apoptosis triggered by immune cells. Defective apoptosome formation. Mutations in caspases, the proteases that execute apoptosis, can impair the apoptotic process. Caspase 3, caspase 8, and caspase 9 are commonly affected in various cancers. Caspases are key regulators of cell death process and are involved in breaking down cellular components. I shall now be discussing about the major aspects of carcinogenesis. It is the process by which normal cells transform into cancer cells. The process is elaborate and quite complex, and therefore, only the key concepts will be told about, which I feel are important for undergraduate students of medical science. Cancer development, or carcinogenesis, is a complex, multi-step process involving various genetic, lifestyle, environmental and other factors. It encompasses the transformation of normal cells into malignant ones through a series of biochemical and molecular events. This process can be broken down into three distinct stages: initiation, promotion, and progression. Carcinogens are substances that can cause cancer. All carcinogens are mutagens, but all mutagens are not necessarily carcinogens. Benzene, tobacco smoke, arsenic, hepatitis B virus and human papillomaviruses, UV rays, etc. Cancer initiation is the genetic hit. It is the first stage of cancer development, where normal cells undergo genetic mutations. These mutations can be caused by various factors such as carcinogens, viruses, or inherited genetic predispositions. The initial mutation affects the DNA of a single cell, leading to changes in cell growth and behavior. Carcinogenesis begins with the initiation phase, where a normal cell undergoes genetic mutations. These mutations can occur in oncogenes, tumor suppressor genes, DNA repair genes, and other relevant genes. Cancer promotion is the stage of clonal expansion and reversible changes. It's the second stage. Promotion involves the clonal expansion of the initiated cell. During this stage, promoters stimulate the proliferation of mutated cells. Such promoters include chemicals, hormones, or chronic inflammation. Unlike initiators, promoters do not directly cause DNA mutations, but enhance the growth of mutated cells, increasing the likelihood of additional genetic alterations. The key points involved in cancer promotion stage include proliferative signals, chronic inflammation, hormonal influence, and cell signaling pathways. Proliferative signals. The promotion phase involves the selective clonal expansion of the initiated cells. This stage is driven by non-genotoxic agents, such as hormones, growth factors, and inflammatory cytokines, which provide proliferative signals without directly causing DNA damage. Chronic inflammation. Persistent inflammation can act as a promoter by creating an environment rich in growth signals and reactive oxygen species, which can further damage DNA and promote cell proliferation. Hormonal influence. Hormones like estrogens and androgens can stimulate the growth of hormone-responsive tissues, increasing the risk of cancer in organs like the breast and prostate. Cell signaling pathways. Activation of pathways like PKC and IMA-PK. IAK promotes cell cycle progression and inhibits apoptosis, aiding in the expansion of mutated cells. Cancer progression is the third stage. It is the stage of genetic instability and malignant transformation. The progression phase is marked by further genetic and epigenetic changes that enhance the malignant potential of the proliferating cells. These changes include additional mutations, chromosomal aberrations, and alterations in gene expression. The key points include inactivation of tumor suppressor genes, activation of oncogenes, and evasion of apoptosis. The mechanisms involved have already been described earlier in this video. Biochemical tumor markers, and biochemical basis of cancer therapy. After completing this section, you will gain an initial understanding of these concepts. Biochemical tumor markers are substances produced by cancer cells or by the body in response to cancer. They can be proteins, hormones, enzymes, or genetic material that are detectable in blood, urine, or tissues and can help in the diagnosis, prognosis, and monitoring of cancer. Some common biochemical tumor markers have been mentioned in this list given alongside. Alpha-fetoprotein, carcinoembryonic antigen. Prostate-specific antigen, cancer antigen 125. Carbohydrate antigen 19-9. Human chorionic gonadotropin, lactate dehydrogenase. And BRCA1 and BRCA2 mutations. Alpha-fetoprotein is primarily used to diagnose and monitor hepatocellular carcinoma and germ cell tumors of the testis and ovary. Alpha-fetoprotein is a glycoprotein normally produced by the fetal liver, yolk sac, and gastrointestinal tract. Elevated levels in adults suggest malignancy. Carcinoembryonic antigen is used to monitor colorectal cancer, and it can also be elevated in breast, pancreatic, gastric, and lung cancers. Carcinoembryonic antigen is a glycoprotein involved in cell adhesion, normally produced during fetal development, but found in low levels in adults. Elevated levels indicate cancer presence and can help monitor treatment response. PSA is used for screening, diagnosing, and monitoring prostate cancer. PSA is a serine protease produced by the prostate gland. Elevated levels can indicate prostate cancer, though benign prostatic hyperplasia and prostatitis can also increase PSA levels. Cancer antigen 125 is primarily used to monitor ovarian cancer, but can also be elevated in other gynecological cancers. CA125 is a high molecular weight glycoprotein produced by mesothelial cells lining the peritoneum, pleura, and pericardium. Elevated levels are associated with malignancy. Carbohydrate antigen 19-9 is used to diagnose and monitor pancreatic cancer and, to a lesser extent, colorectal and gastric cancers. CA19-9 is a sialylated Lewis antigen involved in cell adhesion. Its elevated levels are associated with malignancy and can correlate with tumor burden. Human chorionic gonadotropin is used to diagnose and monitor gestational trophoblastic disease and germ cell tumors. HCG is a glycoprotein hormone produced by the placenta during pregnancy. Its elevation in non-pregnant individuals suggests malignancy. LDH is a non-specific marker used to assess tumor burden and prognosis in various cancers, including lymphoma, leukemia, and melanoma. Lactate dehydrogenase is an enzyme involved in glycolysis. Elevated levels indicate tissue damage and high tumor cell turnover. Genetic testing for BRCA1 and BRCA2 mutations is used to assess the risk of breast and ovarian cancers. BRCA1 and BRCA2 genes encode proteins involved in DNA repair. Mutations in these genes impair DNA repair, leading to increased cancer risk. Biochemical basis of cancer therapy. Cancer therapy aims to selectively target and destroy cancer cells while minimizing damage to normal cells. The biochemical basis of cancer therapy involves exploiting the differences between cancer cells and normal cells. The various types of therapy available include chemotherapy, targeted therapy, epigenetic therapy, hormonal therapy, and immunotherapy. Chemotherapy. Chemotherapeutic agents target rapidly dividing cells by interfering with DNA replication, mitosis, or metabolic pathways. Alkylating agents, such as cyclophosphamide and cisplatin, form cross-links in DNA, preventing replication. The antimetabolite methotrexate inhibits dihydrofolate reductase, blocking DNA synthesis. 5-fluorouracil inhibits thymidylate synthase, impairing DNA synthesis. Mitotic inhibitors like paclitaxel and vincristine disrupt microtubule function, preventing cell division in the process. Some important categories of chemotherapeutic agents include alkylating agents, antimetabolites, and mitotic inhibitors. Targeted therapy. Targeted therapies specifically inhibit molecules involved in cancer cell growth and survival. Tyrosine kinase inhibitor. Imatinib targets the BCR, or BL fusion protein in chronic myelogenous leukemia, the HER2 inhibitor. Trastuzumab targets the HER2 receptor in HER2-positive breast cancer. The VEGF inhibitor. Bevacizumab inhibits vascular endothelial growth factor, reducing angiogenesis. Epigenetic therapy. Epigenetic therapies modify the expression of genes involved in cancer without altering the DNA sequence. DNA methyltransferase inhibitors. Azacitidine and decitabine inhibit DNA methyltransferases, thereby reactivating silenced tumor suppressor genes. Histone deacetylase inhibitors. Vorinostat and romidepsin inhibit histone deacetylases, promoting the expression of genes that induce cell cycle arrest and apoptosis. Hormonal therapy. Hormonal therapies block the effects of hormones that stimulate cancer growth. Selective estrogen receptor modulator, tamoxifen, blocks estrogen receptors in breast cancer. Aromatase inhibitor, onostrozole inhibits aromatase, reducing estrogen production in postmenopausal women. Immunotherapies stimulate the body's immune system to recognize and destroy cancer cells. Checkpoint inhibitors, such as pembrolizumab and nivolumab, block PD-1, PD-L1, enhancing T-cell activity against cancer cells. Chimeric antigen receptor T-cell therapy involves modifying a patient's T-cells to target cancer cells.
Thanks for watching this video. Our next tutorial in the Medical Biochemistry Power Capsule series is titled Immunology. The tutorials presented in the MBPC series are not intended to be a substitute for your textbooks. However, we feel that these tutorials can prove to be very valuable as complementary educational aids and for purpose of rapid revision prior to exams. If you have enjoyed this video, don't forget to hit the like button, subscribe for more exciting content, and share with your friends. And do keep watching our channel to remain ahead in the game.